Good morning, everyone. I am delighted to welcome you all today. Thank you for joining this session. Today, we will be exploring the qualifications of air samplers, a crucial aspect in ensuring air quality. The presentation is titled Dr. S.K. Singh, Ph.D., and focuses on the qualification of air samplers. Over the next few slides, we'll cover the key standards and criteria that define effective air sampling equipment. So, let's get started and delve into this important topic together. Let's take a closer look at the regulatory expectations surrounding air sampler qualification. On this slide, we see references to key standards and guidelines such as EU GMP Annex 1, ISO 14698, USB 1116, and FDA guidance for sterile manufacturing. These documents set the foundation for how we approach microbial monitoring and sampling in clean environments. The main purpose of these regulations is to ensure that our microbial monitoring is reliable, accurate, and efficient. This helps us verify that our sampling methods are performing as expected, which is crucial for maintaining compliance with good manufacturing practices, or GMP standards. Additionally, proper qualification supports our contamination control strategy, what we call CCS, by providing confidence that our clean rooms are effectively monitored. This is vital because it allows us to detect airborne microbial contamination early, validate that our aseptic processing conditions are maintained, and ensure the integrity of our data and, ultimately, the safety of our products. Now, moving to the core question, what exactly is air sampler qualification? It's a documented process where we verify that our air sampling equipment consistently performs according to specific, predetermined standards and regulatory requirements. This process is essential to confirm that our sampling methods are trustworthy and compliant, forming a critical part of our overall quality assurance. Now, let's take a closer look at the typical output we expect from air sampling, measured in CFU per cubic meter, which stands for colony forming units. This helps us quantify the level of microbial contamination in the air. When it comes to air samplers, there are several common types we use. Active air samplers are among the most prevalent, and they work by actively drawing in air to collect samples. Impaction samplers direct air onto a nutrient surface to catch microorganisms. Centrifugal samplers spin the air to separate particles based on density, and filtration samplers pull air through a filter that traps microbes. You might be wondering why qualification of these samplers is so crucial. Well, it's all about ensuring accurate microbial recovery, meaning we're effectively capturing what's truly in the air. Consistent airflow rate is vital because fluctuations can lead to unreliable data. And of course, reliable monitoring results are essential for maintaining a clean environment and ensuring product safety. Now, where do we use these air samplers? They're key in aseptic filling areas, sterile manufacturing zones, and biotechnology laboratories, places where sterile conditions are non-negotiable. They're also used in hospital clean areas and medical device manufacturing, helping us verify that these critical environments stay contamination-free. Finally, the role of air samplers in environmental monitoring cannot be overstated. They are fundamental tools for detecting viable airborne contamination, especially in classified clean room environments. By regularly monitoring the air, we can ensure that the cleanliness standards are maintained, safeguarding both product quality and patient safety. Now, let's take a look at some additional activities that are essential for maintaining the integrity of our air sampler. These include calibration, preventive maintenance, requalification, and change control. Each of these plays a vital role in ensuring the device remains fit for its intended use throughout its operational life cycle. Calibration is about verifying and adjusting the sampler to ensure accurate and reliable performance. Preventive maintenance involves routine checks and servicing to prevent unexpected failures. Requalification is performed periodically or after significant changes to confirm the sampler continues to meet specifications. And finally, change control helps us manage any modifications systematically, ensuring they don't compromise the device's performance. The main objective here is to demonstrate that, Through these activities, the air sampler remains suitable for its purpose from installation through ongoing use. This brings us to the qualification life cycle, which is fundamental to understanding how we validate and maintain our equipment over time. Qualification follows a life cycle approach, encompassing several key stages. These stages are design qualification, or DQ, which confirms that the design meets requirements, installation qualification, or IQ, ensuring the equipment is installed correctly, operational qualification, or OQ, to verify it operates as intended, and performance qualification, or PQ, which confirms it performs reliably in real-world conditions. By following these stages, we ensure continuous validation of the air sampler, maintaining compliance and confidence in its performance throughout its operational life. Now, let's take a closer look at the key parameters we need to consider when selecting an air sampler. First, the sampling volume range is crucial because it determines how much air we can analyze within a given time, impacting the accuracy and reliability of our results. Next, flow rate accuracy is essential to ensure that the volume of air sampled matches our expectations, which directly affects data quality. We also need to consider whether the sampler is compatible with HEPA filters or other sterilization methods, especially in clean room environments where contamination control is vital. Portability and sterilization are other important factors. Ideally, the device should be easy to move around without compromising sterility, and it should be straightforward to sterilize between uses to maintain hygiene standards. To give you an example, the MAS 100NT air sampler is often chosen for grade A and B clean room monitoring because it meets these key parameters effectively. Moving on to design qualification, or DQ. This is a critical step where we verify that the chosen air sampler design truly meets our specific user requirements and intended applications. Activities involved in DQ include assessing the vendor thoroughly to ensure they meet quality standards, reviewing technical specifications in detail, and verifying that the device complies with GMP regulations. We also need to assess material compatibility, ensuring that the sampler's components won't react adversely with the environment or samples, and evaluate its airflow capacity to confirm it can handle the required sampling volumes. All these activities are documented systematically. We prepare a user requirement specification, which captures what we need from the device, and a functional specification that details how it should perform. Additionally, we compile design review reports to record the evaluation process, ensuring everything aligns with our quality and compliance standards. Let's take a closer look at the verification process for the Sartorius MD8 AirScan, focusing on several key aspects. First, we need to verify the serial number, airflow head, and calibration status to ensure everything matches the records. Next, we move on to documentation checks, which are crucial for compliance and traceability. This includes reviewing the user manual, wiring diagrams, calibration certificates, and material certificates. Ensuring these documents are complete and approved confirms that the equipment has been properly documented. The acceptance criteria are straightforward. The equipment must be installed exactly as per the approved drawings, and all documentation should be available and approved before moving forward. During IQ activities, we verify the equipment identification, check utility and power supplies, and confirm that all accessories and components are correctly installed. Reviewing calibration certificates and confirming the availability of SOPs are also essential steps. Finally, the purpose of installation qualification is to verify that the air sampler is installed correctly, following the manufacturer's specifications. This step helps ensure that the equipment is ready for reliable operation and that it meets all quality standards necessary for our process. Let's take a closer look at this example of testing airflow consistency, specifically at 100 liters per minute using a Merck MAS 100 air sampler. When conducting this test, we typically use a few key instruments. First, a calibrated flow meter to ensure accurate measurement of airflow. Second, a tachometer to verify the rotation speed of any moving parts. And third, a stopwatch to keep track of timing during the test. Now, when we talk about acceptance criteria, we're looking for a flow rate that stays within plus or minus 5% of the target, which is 100 L-min in this case. Additionally, we want to see stable airflow throughout the entire operation, meaning no fluctuations that could impact sampling results. Moving on to the operational qualification, or OQ test parameters, these are the specific checks we perform. First, verifying the airflow rate itself to confirm it meets specifications. Then, checking the timer's accuracy, since precise timing is crucial. We also test alarm functionality to ensure alerts activate when parameters are out of range. Sampling volume accuracy is essential to confirm the sampler collects the correct amount of air. And finally, we verify the battery backup to ensure the device can operate or retain data during power interruptions. To clarify, operational qualification is essentially demonstrating that the air sampler performs according to its predefined operational specifications. This step is critical because it confirms the device's reliability before we proceed with routine sampling activities. Overall, these tests and criteria ensure that our sampling process is both accurate and consistent, which is fundamental to maintaining quality and compliance in our environmental monitoring programs. In this slide, I want to walk you through an example of monitoring a Grade B clean room during dynamic conditions, and specifically, how we use active air sampling for this purpose. First, let's look at the key parameters we focus on. Recovery efficiency is crucial because it tells us how well our sampling method retrieves microbes from the air. Repeatability is about ensuring our results are consistent across multiple tests, which helps us trust the data. Environmental suitability refers to whether the sampling method works well under the actual conditions of the clean room, which can sometimes be challenging. Operator reproducibility is important because it confirms that different operators can perform the sampling in a way that yields comparable results. Moving on to the acceptance criteria, we aim for consistent microbial recovery, meaning our sampling method reliably detects what's there without fluctuations. Reproducible sampling results are essential to validate that our process is stable over time. Now, let's consider the PQ activities. These include microbial recovery studies, where we test how effectively microbes are recovered during sampling. We also perform sampling in real operational clean room conditions to see how the method performs in the actual environment. Comparing our results with reference methods helps us validate the accuracy of our approach. Reproducibility testing ensures that the method yields similar results across different tests and operators. Finally, the core definition of performance qualification, or PQ, is that it's a verification process. It confirms that the air sampler performs effectively under real-world operating conditions. This step is vital because it assures us that our sampling process is reliable and suitable for maintaining the integrity of our clean room environment. Now, let's talk about calibration standards. It's essential to use NIST traceable standards to ensure our measurements are accurate and reliable. Additionally, calibration should be performed according to ISO 17025 accreditation, which guarantees that our calibration processes meet international quality standards. When it comes to documentation, we rely on calibration certificates, which provide official proof of calibration labels that are attached to equipment for quick identification, and trend records to monitor calibration performance over time. For example, imagine calibrating a 100 L-min air sampler using a certified digital flow calibrator. That's a typical scenario that highlights how we apply these standards in practice. The purpose of calibration is straightforward. It's to make sure that our airflow and sampling volume measurements are accurate, which is critical for data integrity. Key parameters we focus on include flow rate, sampling time, volume accuracy, and pressure integrity, all of which affect the quality of our sampling results. As for how often we need to calibrate, generally it's recommended every 6 to 12 months, but this can vary depending on our SOPs or the manufacturer's recommendations. Overall, consistent calibration is vital in maintaining the accuracy of our air sampling equipment and ensuring our data is trustworthy. Let's talk about the importance of this step. It's crucial because it ensures that the sampler doesn't damage the microorganisms during collection, which could affect our results. Now, when evaluating the sampler, we look at several parameters. Positive hole correction is important because it accounts for multiple particles passing through the same hole, giving us a more accurate count. Recovery percentage tells us how effectively the sampler recovers microorganisms, and repeatability ensures consistent results across multiple tests. The test procedure involves a few key steps. First, we generate a controlled microbial aerosol. This creates a known amount of microorganisms in the air. Then, we perform air sampling to collect them. After that, we incubate the media to allow any recovered microorganisms to grow. Finally, we compare the colonies we observed to see how many were recovered versus how many were initially present. We often test with common challenge organisms like Staphylococcus aureus, Bacillus subtilis, Aspergillus niger, and Candida albicans. These are representative of the types of microorganisms we might encounter in real-world scenarios. The goal of microbial recovery validation is to verify that the sampler can efficiently recover airborne microorganisms. This is essential because it confirms the sampler's effectiveness and reliability in actual sampling conditions, supporting the overall integrity of our microbial monitoring process. Let's take a moment to look at some key guidelines and expectations around biocontamination control and microbiological monitoring. First, ISO 14698 provides important guidance on biocontamination control and microbiological monitoring practices. It helps establish standardized approaches to minimize contamination risks. Then, there's USP 1116, which offers specific recommendations for microbiological environmental monitoring. This ensures that the environment stays within acceptable microbiological limits, supporting product safety. Moving on to regulatory expectations, the FDA emphasizes data integrity, traceability, and proper calibration and maintenance records. These elements are crucial for maintaining reliable monitoring processes and ensuring compliance. In addition, focus areas such as adherence to SOPs, documentation accuracy, and periodic review and requalification are vital. They help sustain the effectiveness of your environmental controls over time. Finally, the EU GMP Annex 1 sets clear requirements for qualified environmental monitoring systems, especially in Grade A areas. It mandates continuous monitoring and the scientific justification of monitoring methods to ensure robust contamination control. All these standards and expectations connect to the overall theme of establishing a rigorous, compliant, and scientifically justified microbiological monitoring program. They form the backbone of a strong contamination control strategy. Let's now look at some best practices for documentation and electronic systems that are crucial for maintaining compliance and ensuring data integrity. First, good documentation practices are the backbone of a reliable quality system. This includes using approved formats to standardize how information is recorded, which helps ensure clarity and consistency. Traceable records are essential because they allow us to track the history of data and decisions. Controlled revisions are equally important. They prevent unauthorized or accidental changes, maintaining the integrity of our documentation. Data integrity compliance means that all our records are accurate, complete, and protected from tampering. When it comes to electronic systems, features like audit trail functionality are vital. These automatically record every change made to data, providing transparency and accountability. Secure data storage is also critical, ensuring that our information remains safe from loss or unauthorized access. Backup and retrieval systems are necessary so we can recover data quickly if needed, maintaining business continuity. Now, moving on to qualification documentation. These are the documents that verify our equipment and systems are functioning correctly and consistently. Essential documents include the user requirement specification, which defines what we need from a system or process, and the risk assessment, which helps us identify and mitigate potential issues early on. Qualification protocols like IQ, OQ, and PQ guide the testing and validation of equipment, while qualification reports document the results. Calibration certificates confirm that instruments are accurate, and SOPs, standard operating procedures, provide clear instructions for operations. Preventive maintenance records help us keep equipment in optimal condition, reducing downtime and ensuring ongoing compliance. Overall, these practices and documents are fundamental to maintaining a compliant and trustworthy quality management system, supporting consistent product quality and data integrity across all our operations. Now, let's discuss some of the corrective actions we can take to address these issues. Recalibration is essential to ensure our equipment is functioning accurately. SOP retraining helps our operators follow the correct procedures consistently. Equipment servicing is necessary to maintain optimal performance, and requalification testing confirms that our systems meet the required standards after maintenance. Moving on to preventive measures, establishing a routine maintenance schedule is key to preventing many common problems before they occur. Conducting trend analysis allows us to identify patterns and potential issues early on. Periodic verification ensures ongoing accuracy and reliability over time. When we look at qualification challenges, we often encounter issues like flow rate deviations, which can impact the quality of our results. Overloading agar plates can interfere with proper growth, and inconsistent recovery results can lead to unreliable data. Operator handling errors are another common source of variability, and equipment contamination can compromise the integrity of our tests. Understanding the root causes is crucial. Improper calibration can lead to measurement inaccuracies. A damaged perforated head may disrupt flow or sample distribution. Incorrect media handling can affect growth conditions, and poor preventive maintenance can allow small issues to escalate into bigger problems. By addressing these root causes with targeted corrective and preventive actions, we can improve our overall process reliability and ensure consistent, high-quality results. So, as we wrap up, I want to emphasize that the qualification of air samplers is absolutely essential for maintaining reliable environmental monitoring and ensuring compliance with regulatory standards in clean room operations. Without proper qualification, the data we collect might not be trustworthy, which could compromise product quality and patient safety. The qualification process involves 3 critical stages: IQ, OQ, and PQ. Each stage plays a vital role in verifying different aspects of the equipment's performance. IQ, or installation qualification, confirms that the air sampler has been installed correctly. OQ, operating qualification, ensures that it operates within specified parameters during normal use. And PQ, performance qualification, verifies that it performs effectively in the actual environment. Calibration is another key component. It ensures that the airflow measurements are accurate, which is crucial because airflow directly impacts particle and microbial collection. Recovery validation is equally important because it confirms the sampler's ability to reliably collect microbes. This step guarantees that when contamination is present, the sampler can detect it effectively. Proper documentation throughout all these steps supports good manufacturing practice, or GMP, compliance. It provides a clear record that all qualifications and calibrations have been performed correctly, which is essential during audits and inspections. Finally, qualified air samplers significantly enhance contamination control. They give us confidence that our environmental monitoring is accurate and consistent, ultimately helping us maintain a safe and compliant clean environment. So, in summary, thorough qualification, calibration, validation, and documentation are all critical elements that ensure our air sampling processes are robust, reliable, and compliant with industry standards. Now, let's talk about some essential quality assurance practices to keep our air sampling processes reliable and compliant. First, implementing change control is crucial. Whenever we make any adjustments, we need to document and review them carefully to ensure they don't negatively impact our results. Maintaining a qualification matrix helps us keep track of all equipment and personnel qualifications, making sure everything stays up to date. It's also important to review environmental monitoring trends periodically. This way, we can identify any patterns or issues early on. If deviations occur, prompt investigation is key. This helps us address problems before they affect product quality or safety. Moving on to best practices for air sampler qualification, there are some technical points to highlight. Using certified calibration standards ensures our instruments are accurate. Performing periodic requalification confirms ongoing reliability, and conducting microbial recovery studies helps us validate that our samplers can effectively detect microorganisms. Additionally, using validated media and incubation conditions guarantees that our microbial testing is consistent and trustworthy. On the operational side, training personnel regularly is vital. Well-trained staff are our first line of defense against errors. Maintaining equipment cleanliness prevents contamination, and storing sampler accessories properly ensures they remain in good condition. Lastly, monitoring environmental trends continuously allows us to catch potential issues early, maintaining a controlled environment for our sampling activities. All of these practices tie back to our overall goal, ensuring the integrity and quality of our products through rigorous and consistent air sampling protocols. For further support, please feel free to contact us. We're here to help you with any questions or additional information you might need. Remember, we're committed to your success and want to ensure you have all the resources you need to implement what we've discussed today. Thank you so much for your time and attention. We truly appreciate your engagement and look forward to supporting you moving forward.